I now want to talk about the root knot nematodes. This is probably the most important nematode pathogen of all the nematodes. There are over 100 species of this nematode. Every crop plant grown is susceptible to at least one of these. Out of the 100 species, there's probably 10 or 20 that do most of the damage. And not all crops have severe losses, but it's a major problem on vegetables, cotton, soybeans, a lot of ornamental crops. It's important. It's important on turf. Not real important on most grain crops. They're referred to as a root knot nematode because they cause this massive swelling and galling of the infected tissue, shown here, a highly galled cotton root system with a severe infection. This plant would have essentially no yield. More typical symptoms in the field are these cotton plants, where the galling is much more indistinct and difficult. Because of this and because of the presence of the galling on the secondary roots, if a grower who rarely looks at the cotton roots, if he does and he goes into the field and just pulls the plant out of the soil, he's going to strip most of these off. You'll see a nice taproot, you won't see much. If he digs it up, carefully shakes off the soil, maybe even dips it in a bucket of water, then the swellings begin to become apparent. But even the average grower may not notice these swellings as opposed to a severe infection like that. The root knot nematode is very similar to the cyst nematode in that, again, only the second stage juvenile that hatches from the egg is infected. No other stage is infective. So there's only one stage for inoculum. It invades typically the root tip, and it begins feeding on the protoxylem and protophlom, those cells that are in the process of differentiating into xylem and phloem. It feeds on them before they've completed the differentiation, and again induces these to undergo a different developmental path where they become enlarged, highly metabolic active feeding sites. But in this case, we refer to those as giant cells rather than syncytia with cyst nematodes. And I'll talk about the differences between those two types in a minute. Again, once it starts feeding, the worm-like juvenile begins to swell to form the sausage-shaped juveniles. Also, the tissue begins to swell due to hyperplasia and hypertrophy. In the excessive cell division and growth of cells, then you get a gall formation around the nematode. The females remain embedded within this gall, feeding on these enlarged cells until they're reproductively active, and the eggs that are produced, as many as 2,000 per female, are deposited into a jelly-like matrix that is produced by rectal glands and are exposed at the surface of the root. These egg masses are then fall off into the soil. The eggs mature within a couple of weeks, and the cycle begins again. One of the unique things about the sedentary nematodes, the root knot and cyst, is the formation of these giant nerve cells. These are a limited number of 4 or 5 plant cells that the nematodes feed on throughout their entire life cycle to obtain the nutrients for the production of eggs. They're nematode-induced transfer cells that become the permanent feeding sites. Giant cells are noted because they are multinucleated, and each nucleus within the giant cell is polyploid, so that the genome number, copy number in a giant cell is typically 100-fold greater than in a typical diploid cortical cell. So every gene is copied over 100 extra times in these cells. Giant cells induced by the root knot nematodes develop from a single cell, multiple nuclear divisions without cell division. Whereas syncytia in the cyst nematodes develop by the coalescence of adjacent cells in the pericycle or in the dermis. These individual nuclei in syncytia are not polyploid, although the syncytia is multinucleate because of the coalescence of individual cells. Each nuclei remains diploid. An interesting characteristic of these feeding cells is that the inner cell walls are highly invaginated. This greatly increases the surface area of the plasmalemma and increases the amount of area available for active transport of photosynthase into the cell. Hence, they are actually very similar to other plant transfer cells. In both cases, these giant nuclei in large cells are characterized by abnormally high rates of metabolism, including protein and nucleic acid synthesis. These old scanning electron micrographs show some of the cell wall abnormalities of a giant cell. So, in this frame, we have the cavity left by the female nematode, more or less typical cortical cells here. The xylem runs in the center. And these enlarged cells down here, those represent giant cells, as do these up here. If you look closely at these, straight on, you see all of this wall ingrowth material along the inner cell wall, and shown here along this. So the plasmalemma is greatly invaginated as it goes around these ingrowths. Much greater surface area for cell active transport of photosynthase. The nuclear condition is another fascinating part of how these plant parasites change the host. As an example, these are the chromosomes in a normal P nucleus. In the diploid condition, there are 14 chromosomes. This is an example of the chromosomes from a single giant cell nucleus in P. There are over 100 chromosomes in that nucleus, one nucleus. And then you get all kinds of other abnormalities. This is a developing giant cell. It's still elongated. There are, I believe, 8 nuclei in that section. Each one of them is undergoing mitosis at the same time. So it's multinucleate with a coordinated mitotic activity. This is, again, another single giant cell, only part of a single giant cell. It is so large that this mitotic synchrony is beginning to break down, but you have a lot of nuclei in this quadrant undergoing mitosis, but not at this one. You get nuclei that are linked together. And so in some, this is 2 nuclei stained with a Fogan stain. You can just see the chromatin bridge right here between these 2 separate nuclei. And another example of chromatin bridges between nuclei. I believe that this is because the mitotic apparatus, microtubules, etc., is based on 14 chromosomes. You can get enough microtubules of attachment and separate. But when you get a giant cell nucleus with 100 chromosomes, the system no longer works efficiently for the division, complete division of chromosomes during mitosis. And this is just another example of a single giant cell that was dissected from, I think this was broadbean, and the nuclei stained, to give you an idea of the number of nuclei. The invagination not only the cell wall, but the outer cell wall of this feeding site that allowed that single plant cell to provide all the nutrients to that parasite. This is an extremely efficient parasite. It doesn't waste energy on a lot of stuff. It doesn't crawl around. It invades as a juvenile, starts feeding. It becomes a giant egg factory. And all the resources for those eggs come from these transfer cells. One interesting thing about most root knot, the most effective parasites that are in the root knot species, they're parthenogenetic. They don't waste energy on sex. All they do is produce females that feed and produce eggs. And those eggs are then clones of the original female. They produce more females, they produce more eggs, and you can get a 1,000-fold increase in the population in one growing season. And under severe infestation, 2 pictures of modern cotton fields from West Texas, showing the absence of uniform. These 3 rows in the center were treated with nematicide, and in the background, untreated area with severely stunted poor stand. Another field with poor cotton stand due to root knot.